today. So welcome everyone to this CIBERNED webinar series. As you probably know, this is a series that we have been developing for the last 14 or 15 months. And since last January, these are open to everyone which is interested in attending any particular topic. Let me just remind you a couple of general things for smooth development of the webinar. So I would like to ask everyone to keep their micros and cameras off during the presentation so that the streaming goes as smooth as possible. I would also like to say that the webinar today will be recorded, as we usually do, as long as we have the consent for the speaker. After the webinar, the recording will be placed in the CIBERNED YouTube channel. So if everyone is interested afterwards, you can go there and take another look to the presentation of if anyone cannot attend in person in part or totally the presentation, it will be available in just a few days. So as probably you all know, the dynamic of these webinars, there will be a presentation will take around 30 to 35 minutes, and then we'll have some time for questions afterwards. So if anyone is interested in making a question, please go to the chat option in your Teams page and just send your name and affiliation, and I will give you the floor to do the question yourself. So there is no need to write down the question, just your name and affiliation, saying that you're interested in making a question. I think that will be more agile and makes life easier for everyone, and including the presenter. So the seminar, the webinar series is taking place every other week, usually on Thursdays at 3:30. If you're interested in looking at the program for the next few months, it's available at the CIBERNED webpage. I believe until July, we have all the slots already covered. So please go there and then check a look for future webinars. Okay, so after saying that, let's just start, and I would like just to introduce our speaker today. It is a pleasure for me to introduce you all to Montse Solé. Montse got her PhD in neuroscience in the Universidad Autónoma de Barcelona. She did her PhD and the first postdoctoral phase focused on the study of cerebrovascular system dysfunctions in neurodegenerative diseases, specifically Alzheimer's disease, but also on the involvement of these vascular alterations on neurodegeneration, both in AD and stroke pathologies. After a postdoctoral stay in the Universidad Autónoma, she did a stay at the Stanford University in the States, and then moved to the Neurovascular Research Laboratory at the Vall d'Hebron Institute for Research in Barcelona. Since 2019, she is an associate lecturer in biochemistry and molecular biology at the Universidad Autónoma of Barcelona. And in 2020, she joined the cell signaling and apoptosis laboratory led by Dr. Joan Comella at Vall d'Hebron, where she's focused on the characterization of the role of the death receptor antagonist, FAIM-L, in various neurodegenerative disorders, specifically in AD and also in retinal diseases. So with this, she will present today a paper titled Novel Roles of Fast Apoptotic Inhibitory Molecule, FAIM, in Neurodegeneration-Associated Pathology. So thank you very much, Montse, for your willingness to share with us your work today. And the floor is yours. So please share your screen and you can go ahead.
Thank you, Miguel. I'm going to share the screen. And the presentation. Is that okay?
Yep. Okay.
Okay. So thank you, Miguel, for this kind introduction. And I would have the opportunity also to thank the CIBERNED for for allowing me to present the studies that we are performing in the lab at the Institute of Research in Vall d'Hebron Hospital. And obviously, thank you all for attending. So today I'm going to talk about the role of an anti-apoptotic protein FAIM in neurodegeneration. Neurodegeneration is very concisely a process by which neurons lost their function or their structure, and in most of cases, they finally die. Obviously, the loss of neurons has serious consequences in terms of cognitive or motor function, and it accounts for numerous diseases for humans, such as Alzheimer's disease or Parkinson's disease, among others. Despite diverse clinical manifestations, neurodegenerative diseases share common features and mechanisms, and these include, for example, a defective protein quality control and degradation pathways, dysfunctional mitochondrial homeostasis, among others, including neuroinflammation as well. All of them contribute to the death of neurons. But the fact that they are regulated processes means that their knowledge can help us understanding how to modulate them in order to prevent or revert these neurological diseases. The process of cell death is, in most of cases, also a very regulated process. Although different pathways can conduct to cell death, depending probably on the stimulus or also on the cell death, one of the most well-known so far is apoptosis. As most of you may know, there are several apoptotic pathways with multiple proteins involved. The extrinsic pathway, for example, is initiated by the activation of death receptors, and the intrinsic pathway involves the release of apoptotic factors from the mitochondria. Importantly, both pathways are interrelated and converge in the activation of effector caspases, for example, caspase 3 and caspase 7. Even with the importance of maintaining neurons during all our life, due to the low rate of replacement, apoptosis in neurons occurs at the physiological level and represents a very important process during development when neuronal network is established. However, neuronal death becomes a major problem and a clinical challenge in pathological situations, such as in neurodegenerative diseases. So the question is, why to maintain active this crucial molecular pathway in postmitotic neurons? It was discovered that the apoptotic machinery, and in particular the key player in this apoptotic process, that is caspase 3, plays a determinant role in non-apoptotic functions in neurons, which are related with plasticity processes and connections remodulation. In order to avoid an undesired activation of apoptosis, there exists a relative high number of proteins named inhibitors of apoptosis. One example, for example, is SHAP. These are in charge of regulating the activity of this molecular pathway when an excessive activation of this can conduct to the undesired neuronal death. Of particular interest are those exclusively expressed in neurons, which may provide an important chance for the emergence of novel therapies for neurodegenerative diseases. In this regard, in our group, it has been studied for a long time one of these inhibitors of apoptosis, that is FAIM-LONG. This one is an isoform that is particularly expressed in neurons, and which I'm going to focus in my talk today. So, the fast apoptotic inhibitor molecule, or FAIM gene, contains 6 exons and different putative translational start sites. It codifies for 4 isoforms grown so far according to different transcript variants. The main ones are FAIM short and FAIM long, and these are generated by alternative splicing, shown here. FAIM long contains 22 extra amino acids that are in the N-terminal region compared to FAIM short. In addition, there are other 2 minority isoforms that are FAIM short 2A and FAIM long 2A that include the additional exon 2A, here represented, in the respective sequence. These molecules constitute a recently discovered family of proteins that is highly conserved through evolution and is structurally unrelated to other family proteins known. Here you can see the crystallized structure of part of the molecule together with the AlphaFold prediction of the whole molecule, corresponding to the FAIM short sequence. In this case, the 22 extra amino acids of FAIM long would be in this part. A main difference among the principal isoforms, FAIM short and FAIM long, is that while FAIM short is expressed ubiquitously, FAIM long is almost exclusively expressed in neurons. At the functional level, FAIMs were described as inhibitors of apoptosis, but both isoforms do it through different mechanisms. FAIM short was first discovered as a death receptor inhibitor in immune cells. Curiously, FAIM short is able to induce resistance to Fas-mediated apoptosis in non-neuronal cells, but it does not have the same effect in neurons. In neurons, however, it promotes neuronal differentiation and branching through the ERK and the NF-kappa B pathways. By contrast, FAIM long is able to inhibit both Fas and TNF-induced cell death in neurons, and it does this by two different main mechanisms. In one hand, FAIM long directly binds Fas, competing with FAT, and avoiding the activation of caspase 8. On the other hand, FAIM long stabilizes SHAP, the main endogenous inhibitor of the effector caspases. SHAP normally binds caspase 3 and inhibits it. Upon an apoptotic stimulus, SHAP undergoes autoubiquitination and is degraded by the proteasome. Our group has described that FAIM long binding to SHAP prevents its ubiquitination and its subsequent degradation, allowing it to keep caspase 3 inactive. In addition, this system seems to be more complicated, as we have recently uncovered a third partner in this system, that is CIBA1. CIBA1 is able to bind both FAIM long and SHAP and displaces their union, therefore altering the consequent stability of SHAP, inducing its degradation and the consequent activation of caspase 3. Depending on the activation levels or the specific localization of active caspase 3, it can signal through apoptosis or activate these non-apoptotic functions, as I mentioned. These non-apoptotic functions are related to neuronal plasticity and synaptic pruning, and these are processes in which FAIM long is also involved. Neuronal plasticity is required in many forms of learning and memory, and where, through long-term potentiation, or LTP, and long-term depression, or LTD, synaptic adjustments occur. While in LTP, a gain of AMPA receptors is achieved at the surface of the synaptic membranes, LTD implies a decrease in the number of these synaptic AMPA receptors, and consequently, the weakening of the synapse. Caspase 3 is found to be an essential effector in this process, LTD. Another non-apoptotic mechanism of neuronal remodeling is Neurite Branding. This process is essential to sculpt neuronal connections, as it removes excessive or inaccurate projections without resulting in the final death of the neuronal body. The fine-tuned regulation of caspase 3 activity by FAIM long and CIBA1 confers to these proteins a determinant role in the regulation of these processes. Actually, as caspase 3 activity regulator, we have described that FAIM long also plays a role in AMPA receptors internalization. Through in vitro chemical LTD approaches, we demonstrated that FAIM long overexpression, which prevents the caspase 3 activation, totally abrogates after AMPA receptors internalization during LTD processes activated by NMDA treatment. On the other hand, the participation of FAIM long in the regulation of Neurite Branding was also demonstrated. We used dorsal root ganglion neurons placed in campanoid chambers, shown here, which allows the establishment of two separate fluid compartments, one for the cell bodies and the other for the axons. With these systems, axons degenerate in response to a local deprivation of NGF. Interestingly, levels of FAIM long decrease during degeneration in this model, and an overexpression of FAIM long prevents this axonal degeneration. Based on these results, and being FAIM-long a regulator of caspase reactivity, it's feasible to think that an alteration of this regulatory protein could be involved in neurodegenerative diseases. So we studied the levels of FAIM-long in brain tissue of Alzheimer's disease patients, as this is one of the most representative neurodegenerative diseases. We observed that FAIM-long levels decreased both at the mRNA levels and at the protein levels with the severity of the disease, showing very significant lower levels in BRAC6 patients. This decrease was also observed in a mouse model of Alzheimer's disease, the PS1 APP, where FAIM-long significantly decreases at advanced ages. Looking for the cause of this FAIM-long alteration, we treated primary cortical neurons with beta-amyloid aggregates, and we found that in these in vitro conditions, A-beta induces a significant decrease in FAIM-long, but not of FAIM-short, indicating that A-beta could be one possible cause of FAIM-long decrease observed in the AD brain tissue. In addition, FAIM-long was shown to be required for the neuroprotective effect of TNF-alpha against A-beta-induced apoptosis. This was also performed in cortical neurons in vitro. TNF activates pro-survival intracellular pathways, and it's able to prevent the cell death induced by A-beta. However, the blockade shown in the black bars of FAIM-long expression suppresses this TNF protective effect. Thus, the protective effect of TNF against A-beta is dependent on FAIM-long expression. After these results indicating an involvement of FAIM-long in Alzheimer's disease-associated neurodegeneration, we aim to find a good animal model to go in deep in this study. We analyzed different transgenic mouse models, and in contrast to what we expected, we found that mouse models which mostly present A-beta pathology, for example, the 5XFAT or the APP23, did not show FAIM-long reduction. However, mouse models of tauopathy presented a significant loss in FAIM-long, suggesting a more complex situation in the downregulation of FAIM-long rather than the mere involvement of A-beta. In this regard, a sequential relationship between A-beta and tau pathology has been postulated to occur, whereas A-beta aggregation would be the primary stimulus that would cause tau alteration, and tau would be more related with the neuronal death. Based on these results, tau pathology could also be a cause of FAIM loss in vivo. To investigate a possible relationship between FAIM-long and tau, we are now characterizing the FAIM-long loss in the P301S mouse model of tauopathy. Besides, we performed an in vitro approach to identify proteins interacting with FAIM-long. After co-immunoprecipitation and mass spectrometry, tau showed up, among other proteins and biological processes. This predicted interaction was validated by a specific co-immunoprecipitation in vitro, where FAIM-long was shown to co-immunoprecipitate better with tau compared to FAIM-short. We are now following with these studies in order to elucidate the physiopathological meaning of this interaction between tau and FAIM-long. Another strategy that we are using to learn about the role of FAIM-long in neurodegeneration is to study the brain of FAIM knockout mice. So far, 2 different FAIM knockout mice have been created, but one limitation existing for us is that the 2 of them are lacking both FAIM isoforms, FAIM-short and FAIM-long, which limits somehow the association of the effect of a specific effect found to a specific FAIM isoform. Regarding the models, both models are viable and show normal lifespan. In the first model that was developed by Hu and collaborators by homologous recombination in 2009, an increased sensitivity to fast-triggered apoptosis was detected in some peripheral cells. In addition, there were detected some metabolic alterations that might be probably attributed to the FAIM-short isoform, as it functions as a mediator of AKT signaling. However, these metabolic alterations were not reproduced in a second FAIM knockout mouse that was recently generated by Drs. Kaku and Rothstein in 2020. Interestingly, in the second model, the authors detected accumulation of ubiquitinated and aggregated proteins in peripheral organs after performing an in vivo oxidative stress insult with injection of menandione. They confirmed this effect by in vitro experiments using FAIM deficient cells. Protein aggregation, as you know, is one of the hallmarks in neurodegenerative diseases. And in fact, these authors have recently reported that both isoforms of FAIM are able not only to prevent, but also to dissolve pathological aggregates of A-beta in vitro. To explore the possible alterations of FAIM deficiency in neurons, we collaborated with Dr. Huo and Dr. Lam to study the brains of their knockout mouse model. FAIM knockout brains did not show apparent structural abnormalities nor alteration in the morphology and layers of the different brain regions, and they showed absence of apparent apoptotic cell death at any age studied. The expression of the main synaptic proteins in the hippocampus was not altered in FAIM knockout mice, neither neurogenesis or dendritic arborization. Thus, we did not find any relevant brain alteration that could be associated to the absence of FAIM in these animals. However, since besides brain, other organs in the central nervous system can be affected by neurodegeneration, for example, the retina, we next studied this neuronal tissue in these mice. Interestingly, FAIM is highly expressed in retinal tissue, especially in rod photoreceptors. In addition, alterations in FAIM expression have been reported in Norris disease. Mice showing this pathology have an abnormal development of the retina with blindness and report a decrease in FAIM expression. Also, two microRNAs that have been reported to regulate FAIM expression have been associated to different retinal pathologies, such as retinitis pigmentosa and diabetic retinopathy. From these evidences, we evaluated possible neurodegeneration signs in the retina of FAIM cow mouse model of Dr. Huo and collaborators. Very briefly, the retina is a very specialized and organized neuronal tissue that is in charge of processing the phototransduction signals. It's formed by different layers that include nuclear layers, separated by other layers with absence of cell nuclei. In the more superficial part, we find the ganglional cells layer, and going deeper, we find a second layer with bipolar, amacrine, and horizontal cells. More deeply, we find the photoreceptors layer with rods that are more abundant and cones. In addition, retina possesses a specialized glial cell type that is the Muller cells. Since the absence of FAIM was previously associated to increased aggregates of ubiquitinated proteins in peripheral tissues, we first studied whether this phenomenon was also occurring in the retina of FAIM knockout mice, and we analyzed these mice at 2 and 12 months of age. Results, as you can see, showed a significant increase of ubiquitin-positive cells in the internal nuclear layer and in the ganglional cells layer, already from 2 months of age. Bipolar and ganglional cells are the cells that are found in these layers. We then measured whether this ubiquitin accumulation was associated to an increased appearance of markers of inflammation markers. Inflammation is a phenomenon strongly associated with neurodegeneration. And we observed that GFAP, both in mRNA levels or by immunofluorescence, showed a significant increase in this marker, indicating the activation of Muller cells. Retina is a vascularized tissue as well, and similar to the brain, it possesses a blood-retinal barrier that isolates this tissue from the peripheral surrounding. The retinal vascular system contains different vascular levels that irrigate the different retinal layers. As the vascular system can be affected by alterations in the parenchyma, or alternatively, it can happen on the other way around, we explored the blood-retinal barrier integrity in FAIM knockout mouse by measuring the parenchymal extravasation of Evans blue. Results showed increased number and intensity of Evans blue extravasations in FAIM deficient mice, both at 2 and 12 months of age. These alterations were basically found in the superficial vascular plexus that was irrigating the ganglional cells layer, which also showed the highest GFAP signal. We next analyzed whether cell death was increased in these FAIM knockout retinas as a consequence of the FAIM deficiency itself or due to the alterations that we just mentioned. A moderate decrease in the number of photoreceptor rows was detected in the outer nuclear layer, only at advanced stages. That was 18 months old, but not before. No significant differences were observed in ganglional cell layer, and in the same line, a significant number of tunnel-positive cells was detected in the retinas of FAIM knockout mice, also at this age, at 18 months of age, but without representing a massive increase in cell death. However, by OCT micrograph, we could detect a significant decrease in the whole retina thickness, that it was accounted basically for the outer nuclear layer, where photoreceptors are located. In the absence of a massive cell death, we analyzed a possible compensatory signaling pathways that could be active under these conditions. It has been described as an endogenous protective response by which photoreceptors, let's say when they are in trouble, they can induce the activation of Muller cells by the production of endothelin-2. This signal induces the consequent increase in GFAP, that indicates the activation of Muller cells, and the release of FGF2 that accounts for a delay in photoreceptor degeneration. When we analyzed the levels of endothelin-2 in retinas of FAIM knockout mice, we found an increased immunoreactivity in the outer plexiform layer at 12 months of age. The signal was found enclosing the nuclei and following the axonal projections of cone photoreceptors, which are distinguished from rods by the discontinuous heterochromatin, as shown in this diagram. Accordingly, endothelin-2 mRNA and FGF2 mRNA were found significantly increased at 12 months of age. The results suggested that the endothelin-2 survival signaling pathway is activated in these FAIM knockout retinas. Since changes at the signaling level had been found, we next focused on alterations that could occur at the functional level in the absence of FAIM. In collaboration with the laboratories of Dr. Enrique de la Rosa and Dr. Pedro de la Villa in Madrid, we performed in vivo retinal function studies on the 18-month-old mice. We first performed standard electro-retinograms in order to measure the electric activity of the retina in response to a light stimulus. Here you can see a representative image of one typical electro-retinogram. When different intensity lights are applied, the electrical response obtained can give us an idea of which cell types functionally can be altered. In this sense, according to this image, the A wave would correspond to the photoreceptor hyperpolarization, and the B wave represents the bipolar cell's depolarization. Electro-retinogram results showed that the amplitude of both waves was decreased in FAIM knockout mice with increasing intensities of the light stimulus. You can see the FAIM deficient mice in red. These results indicated that there were alterations in the activity of both photoreceptors and their associated bipolar cells. In order to discriminate between the activity of rod and cone photoreceptors, we performed a special protocol that is named flicker. In this protocol, a constant low-intensity light bleaches rod's response, and sequential flashes induce a specific cone response. The absence of differences indicated that the alterations observed in the A wave amplitude are mainly due to alterations in rod photoreceptors instead of cone photoreceptors. In addition, we can measure other parameters in electro-retinograms after bright light flashes exposure. After this, they appear small wavelengths that are named oscillatory potentials, and being the fourth one, or OP4, elicited by retinal ganglion cells. The activity of these cells can also be explored with the stimulation with low luminancies that do not trigger a signal from photoreceptors or bipolar cells, but are able to activate somehow ganglion cells. Analyzing the results corresponding to this data, OP4 and the STRs, we found that in both cases, the activity of ganglion cells is reduced in FAIM knockout retinas. These results therefore indicated that photoreceptors and especially rods, bipolar cells and ganglional cells activity is reduced in retinas of FAIM knockout mouse. In addition to going deep in these functional alterations, we next performed a dark adaptation protocol in which two light flashes are separated by different times. After the reception of a light stimulus, photoreceptors are bleached and they need a time to recover the sensitivity to light and to respond with the same intensity to a second stimulus. If the time between the two light stimuli is not enough to allow the functional recovery of photoreceptors, the amplitude of the second stimulus compared to the first one is smaller, and that's what we measure. Results show that FAIM knockout retinas need more time to recover the light sensitivity, in contrast to wild-type retinas. This result pointed to a possible specific function of FAIM in retinal phototransduction, in addition to a general cellular malfunction that could be consequence of ubiquitin accumulation or inflammation that we have previously observed. So therefore, we next were interested in the phototransduction signaling pathway at the molecular level. To make the long story short, I'm going to focus only in three main actors in the scene, that are rhodopsin, transducin, and arrestin. In the dark, rhodopsin remains inactive. After a light stimulus, rhodopsin is activated and transducin binds rhodopsin, being released the transducin alpha subunit, which activates phosphodiesterase and closes the channel at the plasma membrane, and this causes the photoreceptor's hyperpolarization. After this, a deactivation of the signal needs to be performed to open again the channel and allow the processing of a new light stimulus. In the deactivation process, rhodopsin is phosphorylated, and then arrestin binds phosphorodopsin, allowing rhodopsin deactivation and the return to the basal conditions. Preliminary results analyzing the basal expression of two of these molecules show that both arrestin and transducin are decreased in FAIM knockout retinas, which could be related to a delayed activation of photoreceptors and thus to a worse recovery of a light sensitivity, which was observed before in the functional analysis. We are now working in this line to elucidate the role of FAIM long in this signaling pathway. As I commented before, the main limitation of these results is that the knockout mouse model that we have used presents a lack of both FAIM short and FAIM long isoforms. In order to overpass this limitation, and with the collaboration of Charles River, we have generated by CRISPR technology a novel knockout mouse model with the specific deletion of the FAIM long isoform. With this strategy, we have specifically deleted exon 2B, which is the specific one for FAIM long, while FAIM short expression in theory remains unaltered. We have obtained so far different lines that we recently started breeding, but it has been a few months since we got the animal model, so we still haven't had time to perform any analysis with it. But we trust it will help us elucidating the specific relevance of FAIM long in the neuronal physiopathology, both in tauopathies and in retinal neurodegeneration. So as a summary and future steps, first, we have found that FAIM long is decreased in Alzheimer's disease with special relevance in the tau pathology, and that FAIM long co-immunoprecipitates with tau. Therefore, our aim now is to study the relationship between FAIM long and tau, and to evaluate the possible role of FAIM long in tauopathies. Second, we have described that FAIM knockout mice show a neurodegenerative pattern in the retina, with accumulation of ubiquitinated proteins, glial activation, and moderate photoreceptor cell death, as well as functional alterations in the photoreceptors, bipolar and ganglion cells activity. So our next step in this part is to elucidate the role of FAIM long in the phototransduction signaling pathway. And in addition, of course, we are going to focus in the characterization of the new specific FAIM long knockout mouse model, specifically focusing in neuronal and retinal degeneration. And just to finish, let me thank all the members of the group. And I would also like to highlight that the first studies that I presented are the result of the work of several past members from the lab. and the projects ongoing that I showed are being conducted by Raquel Vadillos, in charge of the Apoptosis project, and by Ana Martinez Cires, in charge of the project on retinal physiopathology. And I would like also to thank our collaborators and funders, and of course, all of you for listening to this talk. Thank you very much.
All right, thank you very much, Montse. So the paper is now open for discussion. So let me remind you that if you want to make a question to Montse, please do so by using the chat option in your screen, just by stating your name and your interest in making a question. There is no need to write down the questions, since I just give the floor to anyone interested. Just put your name and your affiliation, and that's it. So in the while we wait for the first questions to arise, let me remind everyone that this webinar has been and is being recorded, and it will be made available to everyone on the YouTube channel from CIBERNED. So you can check if you arrive later, you can check the whole webinar in there. Also, if you're interested in future webinars, our program for the next few months is already available at the CIBERNED webpage. And so you can go there and check the upcoming webinars from now until July, I believe. So I have a hand being raised here by Ignacio Torres Alemán. So Ignacio, if you want to make a question, please go ahead.
Yes, thank you. Thank you very much for your presentation. I came a little bit late, so maybe I missed it, but you don't see any other alteration, gross alteration in the brain of this mutant?
No, that's correct. We have looked for the different layers organization, excessive cell death, neurogenesis, dendritic arborization, and the main synaptic proteins, among other things, and nothing was significant.
Well, it's quite intriguing. Thank you. Thank you very much.
All right, thank you, Ignacio and Montse. Any other question? Okay, let me make a quick question, Montse, if I may. I was also interested on the interaction that you show on the FAIM with tau. Mm-hmm. Have you used whole cell lysates to do that immunoprecipitation experiments, or have you used tissue lysates? Or
what we do is to overexpress both proteins and then do the immunoprecipitation. We have tried several times with lysates, and so far the protocol that we are using seems to not be working very well for tissues. So we keep trying.
Okay. Have you checked whether that tau bound to FAIM is somehow phosphorylated or phosphorylation or modified?
We don't know. We don't know. Okay. But this is something that we are working in to know whether the phosphorylation affects the binding, or even to compare. We are trying to compare with mutant tau that in theoretically is more phosphorylated, to see if we have a different interaction. And also with FAIM short, because although we have checked that at the basal situation, it seems to be less interaction with FAIM short than with FAIM long. It's something that we also need to check with the modifications of tau.
All right, thank you. All right, any other question? I don't see any one raising hands or sending any message through the chat, but we can wait for 1 or 2 minutes just in case someone else is interested in making a question. Okay. Apparently there are no more questions. So this is your last chance to make a question before we close the session then. Anyone?
Well, I must say that we are not experts in tau pathology or in retina, so it's something that we have just find on the way. So if it's anyone, and if there is anyone here that is an expert on these two issues and wants to make us suggestions or whatever, or collaborations, we are open, of course, so feel free to contact us.
All right, thank you very much. So I don't receive any other questions, so I think it's then time to close the session. Not before thanking Montse once again for her willingness to share her work with us. Thank you everyone for attending. And just let me remind you that we'll have the next webinar in 2 weeks' time, always on Thursdays at 3:30 p.m. So thank you, Montse.
You're welcome.
Thank you everyone, and have a great day. Bye-bye.
Bye.